Hello and welcome to this course. I am Dr. Sriram Kumar. Today we are going to learn biological methods of nanomaterials synthesis. This is very important methods of nanomaterials synthesis. So before starting to this topic, we have some important questions in biological method. The first question comes here is what biological system can be used to synthesize nanomaterials? So here in the biological system means what kind of this material we can use, what kind of the materials maybe we can use bacteria, maybe we can use fungi, maybe we can use yeast, or maybe apart from these bacteria, fungi, yeast, can we use the plant-based material, plant-based materials? So these are the questions. So in biological system, which material is best for synthesizing nanomaterial? And the second important question is what is the biochemical process leading to nanoparticle formation? So we need to understand why this biological system makes nanomaterial metal metal ion to the metal zero or M0, and that leads to the formation of nanomaterials. So we need to understand the biochemical process behind this kind of the reduction process. So it is kind of reduction process. And we have third important question is about if we use the biological system, can we control nanomaterials size? Or maybe can we control the monodispersity? So whenever we synthesize nanomaterials, we have kind of this two kind of the dispersity. One is polydispersity, and the second is monodispersity. So when we will synthesize nanomaterial, if we have nanomaterial with the minimum, for example, if we have here the size of the nanomaterial, so suppose if we have one kind of things here, and if we have the other kind of things here is if we have really, so here the second is kind of the monodispersity because that has the less error and that has the maximum intense peak at one wavelength. So this kind of the things is called the monodispersity. However, if we have this broader peak there, so that contributes from the polydispersity. That means you don't have the control over the particle size. You have the distributions maybe with the large distributions. So here, if you measure nanometer size with the intensity here, intensity, we will get this kind of the distribution curve. And based on that distribution curve, we will know, okay, whether we have the monodispersity or maybe the polydispersity kind of the nanomaterial. And we have another important question is how about nanoparticle shape modulation? So can we control the shape and size of the nanomaterial? So that are this very important question if we are opting biological methods. So now let's understand which kind of the system we can use in nanomaterial synthesis. So in this method, we can use either bacteria, maybe fungi, algae, yeast, and the viruses. So we can use these kind of biological systems for nanomaterial synthesis. So either we can use two kind of the mechanism. One is the intracellular kind of the mechanism, and other is the extracellular kind of the mechanism. So we are going to learn what are the difference and what are the intracellular and extracellular mechanism there. So what happens? So if we extract the important chemicals from the bacteria, fungi, algae, yeast, and viruses, for example, if we can extract protein, important protein, maybe polysaccharides, nitrate reductase, or maybe coenzyme. So if we extract these important chemicals from bacteria, fungi, or the other biological system, so these kind of the materials, these are called natural reducing agent, reducing agent. So either we can use protein, polysaccharides, nitrate, or maybe the enzyme. So these are the natural reducing agents. So that converts metal charge to the M0, and that's why we'll get nanoparticle. So this is the kind of the things. So what happens? Either we can extract this chemical intracellularly or maybe the extracellularly. And when we add the here metal and solution there, what happens? So that process happens and we will get the kind of the nanoparticle there. So based on the kind of the capping agent, surfactant, or maybe the shape of this bacteria, we can control here what would be the shape of the nanomaterials. Maybe we will get nano rod, maybe nanovirus, nanoconjugates, or maybe the nanotubes. Okay, so if we use here's bacteria, so if we can either process extracellular kind of the mechanism or maybe the intracellular kind of the mechanism, and for from that bacteria we can develop gold nanoparticle, iron nanoparticle, the kind of silver nanoparticle, and a different kind of the nanoparticle. We can also consider the fungi and the yeast, and these two biological sample system also undergo in the extracellular or maybe the intracellular kind of the mechanism. So we can use for the bacteria and the viruses both kind of the mechanism. And for the plant and the plant extract, we should go for the extracellular kind of the mechanism. So using bacteria, fungi, and yeast and the plant kind of extract, we can develop important gold nanomaterials. So this process is called kind of this green approach. Green approach of nanomaterial synthesis. So if we follow this kind of this mechanism, then we are reducing waste to the mother nature. Okay, so if you want to understand this basic mechanism behind this biological system, so whenever we extract some important chemicals, maybe we can extract the protein, peptides, enzymes there. So these kind of the systems, so these are called the natural reducing agent. So if we extract this one and if we add this metal solution there, what happens? That will reduces into the M0 and we'll get the kind of the nanomaterial. So we will understand this mechanism in more details. So here, the biological sample or the extract from the biological sample, there's just two important role. One is that it acts as the natural reducing agent, natural reducing agent, and the second is the stabilizing agent. Maybe we can say is the capping agent, capping agent. So if we use the capping agent on the nanomaterials, so that capping agent or maybe stabilizing agent keeps stabilizing the nanomaterial in the solution phase. So if you have this kind of the capping agent here, so because of this capping agent or maybe the stabilizing agent, there is kind of the hydrophobic interactions, and because of that, there is the kind of the repulsions happens. Uh, and because of that, that don't allow to settle down this kind of the nanomaterials in the solution. I already explained this kind of the stabilizing effect of stabilizing agent in the stability of nanomaterials in the solution. So you can consider that lecture to get more information about how the stabilizing agent and capping agent stabilizes the nanomaterials. Okay, so now we have big question, and very important question is, why do organisms synthesize nanomaterials? So we can understand. So for that, we can understand that this kind of the nanomaterial synthesis by the biological organism is because of the detoxification effect. So we can understand that detoxification process by just considering, suppose in your house someone enters and you don't know that person, what will you do? You will take some action, depends on the type of the person that will enter to your house. So first, so the action taken by you, maybe it's called the kind of the detoxification process, where you prevent yourself from the foreign particles or maybe the person that is entering in your home. So here, that similar things happens here. If you have this here's bacteria, and if any metal ion that enters in this cell, what happens? That will prevent or maybe minimize the effect of the metal ions toxicity in the cell by taking some kind of the action, and that preventing these one the metal ions within the cell is called the detoxification. So that detoxification process can be happens at maybe the kind of the four types of the detoxification. So let's understand the first thing is the modification in the cellular transport mechanism to resist entry of the toxicon into the cell. So one kind of the mechanism happens is that whenever any metal ions try to enter within a cell, what happens? So this bacteria has some kind of the channel there uh, in the cell membrane or maybe the cell wall. What happens? There is the kind of the channel. So what happens? So that will change the mechanism to enter these metal ions within a cell. So that is kind of called the cellular transport mechanism. So that will change that kind of the mechanism to resist entry of this kind of the toxic metal ions within the cell. So this is one kind of the action bacteria or with biological sample system takes. And the second is the kind of the action is the sequestration of the toxic species within the cell or outside the cell. Intracellular sequestration or the extracellular sequestration. So let's understand what is this kind of this things is. Suppose somehow if the metal ions try to enter, and if you don't able to resist the entry of this one, so what happens? You may take this two kind of the action here. So what happens? You can make these metal ions to the less toxic and store either the outside of the cell, or maybe you can store within a cell in the form of the, so you store, so that bacteria store that metal ions in less toxic form. What happens? So cell takes the action and convert that toxic metal ion into the less toxic form and store maybe the inside, that is called the intracellular sequestration, or maybe outside of cell, and that is called the extracellular sequestration. So till now we understand two processes. One is that cell resists the entry by this changing the cellular transport mechanism. And the second is about the storing of that toxic metal ion in the form of the less toxic form, either within the cell or maybe the outside of the cell. And we have the third important action by this biological system is that activation of energy dependent flux pathways to eliminate the toxic species. So in this particular case, what happens if metal ion enters here? So what happens? So that cell takes the kind of the ATP pathways, some kind of the ATP pathways to throw out this kind of the toxic metal ions within the cell, and that is called the energy dependent efflux pathways, to just kick out this kind of this toxic metal ions within a cell. And we have another important kind of the action is that enzyme catalyzed oxidation or reduction of toxic species to the less toxic form. And that fourth step is about that cell may have the kind of the particular kind of the enzyme which can convert that toxic metal ions into the less toxic metal ion by the either the oxidation process or maybe the reduction process. So here, what happens? So we have metal ion. So what happens? So this kind of the conversions maybe happens on the cell wall or maybe the cell membrane. And there is another kind of the mechanism happens if somehow that metal ions enters into the cell. What happens? So because of that enzyme present in the cell cytoplasm, what happens? So that converts into the metal ion and then it's kicked out from that cell. So what happens? What either this enzyme catalyzed reactions happens on the cell wall or maybe the cell membrane. That depends on the types of the cell we are using. And the second is kind of the we have here that enzymes may present within the cytoplasm and that converts the metal ions into the metal. And these are the process of the detoxification by biological sample. So this point is very important to understand the wise biological system, maybe the organism synthesize nanoparticles. So this is all about the kind of the actions. Okay, so now let's understand the types of those cells. So it is just for the refreshing. Maybe you might forget about the kind of the cell. So we have the two kind of the cell. One is the prokaryotic cell, and another is a kind of the eukaryotic cell. So if you just say the difference in the prokaryotic cell, so in first case, there is no nucleus. So in the prokaryotic cell, there is no such kind of the nucleus present here. However, in eukaryotic cell, we have here such kind of the nucleus present. And in eukaryotic cell, so again, it has the two kind of the cell. So that may one kind of cell may have kind of the cell wall. Maybe that has a cell wall, and maybe the other kind of the cell they only have the cell membrane. So here eukaryotic cell don't have this nucleus. However, the eukaryotic cell have nucleus, and if we for the classify this kind of the cell, then one kind of the cell may have the cell wall, and other kind of cell have only the cell membrane. And if you just understand, so within the cell membrane, so it is the kind of the cell membrane. So in the cell membrane, we have here the nucleus. So within the kind of the cell membrane, we have the cytoplasm, and within the cytoplasm, we have the kind of nucleus, and we have the kind of the floors here, the mitochondria, the endoplasmic reticulum, plasma. So these are the things as present in the cytoplasm. Okay, so we understand till now that why organism converts metal ions into the nanoparticle. Second is that we understand that the types of the cell. Okay, now let's understand this mechanism. So there's synthesis of nanoparticles by bacteria as is two kind of the mechanism. One is the intracellular mechanism, and the second is the extracellular mechanism. So now let's understand intracellular and extracellular mechanism. So every kind of cell has a kind of, here is the charge transporter or maybe the ion transporter. We have an transporter. So what happens through that ion transporter? We have here Mn plus charge there. So through that ion transporter, what happens that so that will enter inside the cell, and that is called the so this portion is called the cytoplasm. Cytoplasm. Okay. So what happens in the cytoplasm? We have one kind of this enzyme. So what happens here? So that enzyme reduces metal ions into the M0, and here, so that enzyme gets oxidized. What happens? So this enzyme reduces metal M plus 2 to M0. So what happens? There is kind of the reduction process happens. And then in second case, there is a kind of the oxidation process happens. And this all process happens within the cytoplasm because of the presence of the particular kind of the enzyme. And we have the second kind of the mechanism is the extracellular mechanism. So in this mechanism, that kind of the enzymes is present on the cell wall or maybe the cell membrane. So it depends on the kind of the cell we are using. So suppose that kind of the enzyme is present on the cell membrane. So again, what happens? We have here this metal ion. So because of that enzyme, so that converts metal ion into the metal zero form. And then for there's a development of the nanoparticles. And so this process is kind of, so in that enzymatic process is kind of a cyclic process, and we will get the kind of the nanomaterials synthesis outside of the cell, or maybe the cell wall that absorb or cell membrane that absorb that nanoparticle on the surface there. So so types of the mechanism we that bacteria either undergo the intracellular mechanism or maybe the extracellular kind of the mechanism. It depends on the that enzyme is present on the cell membrane or maybe in the cytoplasm. Okay, now let's understand with a specific example then how this kind of the mechanism converts or synthesize nanomaterials. So now let's understand with the example of the zinc oxide material. So we have cell here, and that cell, so if we culture bacteria, and in that growth, so if we culture the bacteria here, what happens? So in the intracellular mechanism is that first we need to culture bacteria, right? And then second case is that we should always use bacteria in the growth phase. So bacteria in growth phase. Right? So when we culture the bacteria, and the second is that we need to always use that bacteria in the growth phase conditions. And in that case, when we achieve that in the growth phase of this bacteria, what happens in third step is that we need to add metal ion. Right? So in that case, if we add the metal ions, what happens? So if we add the metal ions, so that metal ions, so because of the electrostatic interaction between that plus charge and negative charge on the cell membrane, so there is kind of that electrostatic interaction happens, and because of that, what happens? And there is kind of the here the ion channel is there on the cell membrane. So because of that, so that enters inside the cell membrane. And once if that enters inside the cell membrane, so if you have this metal ion plus there, and there is kind of the enzyme is present there that is called NADH. So that enzyme reduces that metal ion, so Zn plus 2 to Zn. So what happens? We have this kind of reactions happens here. Zn2 to Zn and 2 electron here. So that reduces, sorry, I made mistake there. So here, so that zinc plus 2 gets reduced into the zinc in the presence of this enzyme, NADH, and that oxidizes itself into the NAD plus. So because of that enzyme present in the cytoplasm, we will have the kind of the nucleation process happens. And because of the nucleation process for the happens, there is a kind of the growth of that nanomaterials. And interesting thing is that within this cytoplasm, we will have the different kind of the amino acids. For example, the cysteine, tyrosine, and tryptophan. So this kind of those amino acids stabilizes that zinc oxide nanomaterials, and that makes the stable form of this nanomaterial. So in intracellular synthesis of nanomaterials, so what is happening? First, we need to culture bacteria and use the growth phase of the bacteria. And then after, we need to add in that culture metal, metal ion there. So because of this mechanism, what happens that metal ions gets transported inside the cytoplasm. And because of this mechanism, and we have the kind of the enzyme that is the NADH. So that enzyme reduces that metal ions into the metal, and enzyme gets itself oxidized into the NAD plus. So this kind of the mechanism happens within the cytoplasm. And once we get the kind of the nucleation process happens, so there is a further growth happens of that nanomaterial, and finally we get the kind of nanomaterials, and these nanomaterials are stabilized by the stabilizing agent, and these are called the stabilizing agent, stabilizing agent, kind of the cysteine, tyrosine, and the tryptophan kind of the amino acids. Now let's understand what is happening in the extracellular synthesis of nanomaterial. So similarly, what we can do again. First, the first step is that we need to cultures particular kind of organisms, bacteria, right? And in the second case, when we culture the bacteria, we need to wait, culture bacteria. And the second is that grow bacteria up to growth stage, and then after what happens? Then after we need to add metal ions. What happens if we add the metal ions? So because of the presence of the enzyme there, so in the extracellular mechanism, so this kind of the enzyme is already present on the cell membrane. So whenever that kind of the metal charge gets interacted with the enzyme, what happens? So we'll have here, so we have zinc plus 2, and when that interact with this enzyme, what happens? So there is the reductions happen, and because of that reduction, so that is kept by the kind of this enzyme there, and we'll have the nucleation process starts. And if that nucleation process happens, and then further growth of the nanomaterials happen there, the here further growth, and finally we get kind of this aggregates on the surface of this bacteria. And here again, these are called the capping agent that is present in the amino acids. And here that capping agents are the kind of the cysteine or tryptophan or maybe the tyrosine kind of the amino acids works as the kind of the capping agent there. So there is only one difference between the intracellular and extracellular kind of the mechanism is that one process happens in the cytoplasm and the second kind of mechanism happens, extracellular mechanism happens on the cell membrane. And so this kind of the reactions happen by the intelligence of the enzyme. NADH plus in kind of the enzyme. So we have the NADH enzyme, and that's gas oxidized NAD plus. So if we just need to understand the kind of the enzyme and what is the reactions happens, and we, in presence of that enzyme, we'll get the nanomaterial. So if you want to get the difference between the extracellular mechanism and intracellular mechanism, so let's understand what are the differences. So in the extracellular mechanism, generation of extracellular reductase in the cell-free supernatant. That means these kind of the reactions happen outside of the cell. So when you grow, culture the bacteria, and if we add that metal ion, we will have that nanomaterial outside of the cell. So that material is not the inside of the cell. So we can easily extract these nanomaterial. However, in the extracellular intercellular synthesis, so that metal ions is present within the cell membrane or maybe the cell wall, depends on the kind of the cell we are using. And the second kind of the difference is that enzymatic reactions happens within this cell-free supernatant. So what happens? That enzymes reactions happen on the surface, and because of that, we will have that reactions in the supernatant. However, that enzymatic reactions are metal within the cell wall. And the third kind of things is the formation of nanomaterials. However, in the kind of intracellular kind of mechanism, you will have the maybe nanocluster formation or maybe the complicated aggregates. And then further, so we can characterize that the nanomaterials if once we get synthesis by the either extracellular kind of mechanism or maybe the intracellular mechanism. So till now, we understand very key point is that why is biological sample make nanomaterial? Why that reduces metal ion into the nanomaterials? Second is that we understand the mechanism of the nanomaterial synthesis by the bacteria, and that was the intracellular mechanism and the extracellular mechanism. Now, let's understand what are the key parameters we can control to develop the nanomaterials using the biological sample or biological system. So these parameters are useful and valid for all kind of those biological samples. So this strategy is useful for all biological systems. It is not only for the bacteria, but it's for the all kind of the biological systems. So now let's understand how biological sample affects the nanomaterial synthesis. So if you don't use kind of this bacteria as a template, or maybe the niche as this bacteria, or maybe any kind of the biological system, so in that case, we will have the large distribution of the here nanomaterials. So here we have large distribution of nanomaterials. Right. So what happened here? There is large distribution of the nanomaterial size, and we have the kind of those polydispersed nanoparticles. Polydispersed nanoparticles that there's the large distribution. At the same time, we don't have the control over the nanomaterial synthesis, so we will have the kind of the heterogeneity kind of the things here. So whenever we use the biological system, so if if you are planning to use the biological system for nanomaterial synthesis, so we need to consider these important parameters. And the first important parameter is about this pH. Here, we need to control the pH of kind of the growth of this biological sample. So it depends on the kind of the bacteria or this biological sample used. So that pH depends on biological sample. Biological sample. Because some bacteria prefer in acidic condition and some bacteria prefer basic condition. So it depends. If bacteria that prefers in acidic condition, in that case, the low pH prefers the faster growth of nanomaterial, and we have the very much control over the particle size. And if that bacteria metabolizes and gives in the basic pH, in that case, that bacteria will give the good nanomaterial control over the pH, maybe the pH 9 to 10. So it depends on the kind of this bacteria sample, biological sample we are using. Second important parameter is kind of the temperature. So generally, if you want to understand the temperature difference, we have here, if you want to understand temperature, maybe if we have the 10 degrees Celsius, maybe 25 degrees Celsius, and maybe 37 degrees Celsius. So if we consider the three temperature, so in that three temperature, based on the research, it is found that there is the smaller particle size at. So here, what happens? If you increase the temperature, that means you have the smaller particle size. So here, if you increase temperature, a small, large size. So at 37 degrees Celsius, you have the smaller size, and at the 10 degrees Celsius, we have the large size of nanomaterial. So it is because of that, at high temperature, what happens? So what is happening at the high temperature? So what is happening is that there is kind of the large high metabolic activity. And because of that metabolic activities, you have the large amount of enzyme there. And because of that large amount of enzyme, what happens? There is the fast nucleation process. If you have the fast nucleation process and the high diffusion, so that leads to, and so what happens? So that leads to the small size of nanomaterials. So these are the things is happening if you use high temperature as compared to the low temperature. So these are the things we need to control at what temperature we should develop or the synthesize nanomaterial. And we have the other important kind of the parameters, that is the metal concentration. And most important thing here is that cells is we there is the huge effect of the cell is. So here cell is very important parameters. Now let's understand how the cell is happens. So if we have two kind of the cell is, maybe we have the growth stage, or maybe the we have the mature state. Not stage. So cell is may have the growth stage and maybe the mature state. And for the nanomaterial synthesis, we will have here growth stage preferred for nanomaterials synthesis. So here we need to use the growth phase. Because of that stage, there is huge high metabolic activity of the bacteria there, and because of that we have the that kinetics of the growth nanomaterials is very high, and we'll get the high output of that nanomaterial synthesis if we use the growth stage as compared to the mature state. And we have the other important parameters, that's reaction times. So that reaction times depends on the kind of the cell you are using and whether you are using short time, long time. We have another kind of the parameter, maybe the kind of irradiation, maybe the stirring process, and what is the diffusions of the metal ions to the bacteria extracellularly or maybe intracellularly. So these are the parameters if we control. So in that case, we will have very much control over the nanoparticle. So in this case, we will get the smaller size first as compared to the before, as compared to the larger particle size. And the second time, we will get the high yield of the nanoparticle. And third important point is that we will get here this monodispersed kind of nanoparticle. So here the monodispersed means that the particle size of that nanomaterials is very much similar as compared to the first one. In the first one, you will have here the large distribution. However, in this case, you will have here very kind of the short distribution, very fine distribution of the nanoparticles. So this kind of the parameters are applicable for all kind of the biological samples or maybe the systems. So if you want to further study this kind of the effect or those parameters on the synthesis of nanomaterials, you may take the references from here. Maybe so in this is kind of this microbial production of the gold nanoparticle, and that so you can just go there and read in details. And if you want to understand the optimization of the extracellular biosynthesis of the silver nanoparticle by this kind of bacteria, you go there and read in details. And another kind of the things is the here this biosynthesis, purification, and characterization of silver nanoparticles. So this one is the silver nanoparticle. This is also silver nanoparticle, and first one is the gold nanoparticle. So if you want to go study, learn more details, go there and study there. So I put this table for your reference if you are looking for reference, or maybe if you are planning to synthesize that nanomaterial using the bacteria, maybe the fungus as a source material. So here, for example, if you use the different kind of this bacteria, and based on that we can control this morphology of that nanoparticle, and we can also use a different kind of the mechanism, maybe the intracellular or maybe the extracellular kind of the mechanism there, and based on that we will have the distribution over the particle size. For example, if you use the E. coli kind of the bacteria, so we will get kind of this triangular nanoparticles shape, and that has the particle size 10 to 20 nanometers, and using the extracellular kind of the synthesis process. Similarly, if you want to use Pseudomonas aeruginosa, so in this case, we'll get kind of the spherical nanoparticle. We'll have the particle size. So it has a 5 to 30 days. That means the maybe the distribution of large distribution of the particle size here, because that is from the 5 to 30 nanometer. So based on the biological system, we can control the two parameters. One is the morphology, one is the particle size, and the dispersity, maybe the monodispersity or maybe the heterodispersity kind of the material. And if you're looking for kind of the fungus as a source, you can use the Fusarium oxysporum. So in this case, you get either spherical or maybe the triangular nanoparticle, and you can control the particle size, maybe 8 to 40 nanometer, using the intracellular kind of the mechanism. And if you are looking to other kind of, we have the rhizopus or EAs. So it depends on the kind of the we will have the rods, triangle, or maybe the hexagonal kind of nanoparticles, and we have very much control over the particle size, maybe 9 to 10 nanometer. So these are the things we can use if you are looking for the algae. So in algae, so this we can use the kind of the extracellular kind of the mechanism. And if you're looking the plant kind of the source, in that case, we can use kind of extracellular mechanism. So in algae and the plant kind of the system, we should go for the extracellular mechanism. And for this bacteria and the fungi, we can either use maybe the extracellular, maybe the intracellular kind of mechanism. If you are looking for more details, so you should go there and read in more details about this kind of the synthesis of nanomaterial. So till now we understand the basic understanding about the synthesis of the nanomaterial by the bacteria and the control over the nanomaterials using the different parameters. For example, we can control by temperature, we can control by pH, maybe we can control over the kind of reaction, maybe the cell is, whether we can, so either using we should always use growth phase of bacteria culture for synthesis of nanomaterials to get the high output of the yield. So these are the things we need to learn. And if we want to understand and take the example of the nanomaterial synthesis using the fungi, so let's understand the synthesis of the nanomaterial by the fungi. So before understanding the mechanism, we should think about, okay, why should consider fungi as compared to the bacteria, and which method is better? Is a fungus-based synthesis, biosynthesis, or maybe the bacteria-based biosynthesis? Okay, so let's understand with the one by one. So first point is that fungus can be used either for the intracellular mechanism or maybe the extracellular mechanism, as so same thing happens for this bacteria as well. And the second is kind of this is the cost effective, because the growth and culture of the fungus is easy as compared to the bacteria, so that makes overall the cost effective process. And the third point is that the metal accumulations capability is better in fungus as compared to bacteria. So here this metal accumulation capability may be either the intracellular kind of things or maybe the extracellular kind of things. And third and the fourth important point is that that fungi has high production of the enzyme for the reduction of the metal. So if we have very large, high concentration, maybe the large production of the enzyme. So in that case, so here, if we have metal ion, so this process becomes more efficient if we have the high production of that particular kind of the enzyme. And the next point is about the effective secretary of the enzyme. So all kind of the fungus has higher metabolic activity as compared to the bacteria, and that's why the fungus grow very at very high rate as compared to the bacteria, and because of that, we will have the high concentration enzyme present, and because of that enzyme, we can use that enzyme for the extracellular kind of the nanomaterial synthesis. And the second point, next point is about the production of the large biomass. So based on the biomass and this metal ion concentration, we can control the overall the particle size and morphology. Next point is about the eco-friendly approach and the variety of nanomaterials can be produced here. So here, the advantages of fungus as compared to this bacteria, we can consider, with these 2 to 3 points. First important point is about that. So this is the very important point is about it secretes high concentration of the enzyme, so we can use extracellularly efficiently. Second, B point is that so the production of the enzyme is very high as compared to the bacteria, so we can use as efficiently. And the third important point is that about those accumulation of that metal within the cell, maybe they're the surfaces. And that's why we should choose fungus kind of the biological systems for the nanomaterial synthesis as compared to this bacterial system. So if you want to understand the synthesis of nanomaterial by the fungus, so again, in the fungus, we have the two kind of the mechanism. One is the intracellular kind of the mechanism, and the second is the extracellular mechanism. So the same thing happens here as we understand in this bacteria. So here, what is happening? So if you want to understand the extracellular mechanism, so this kind of the enzyme is present at the surface here. So what happens? If we have here's metal ion, so what happens? So this enzyme gets oxidized in this form, and if we have here this metal M plus, what happens? So that gets reduced into the M0. So similar things happens here for this gold nanoparticle. So at the surface of the fungus, we have that enzyme there. So because of that enzyme, that gets oxidized, and there is the electron, there is electron and here, so this gold plus 3 metal ion gets reduced to gold 0, and there is formation of this gold nanoparticle, that is capped nanoparticle. Similar things happens inside the cell, where what happens? So this process happens in the cytoplasm as compared to the cell membrane. And we have, so if you want to understand what is happening there, so if we have culture of fungus, what happens? So if we just take the culture of the growth phase of, so this is kind of growth phase of fungi culture. Okay, so if we take the growth phase of the fungus, and if we add here this metal ions, what happens? So they may be two kind of the mechanism happens. One kind of the mechanism is that metal ions enter inside the cell, where so in the cytoplasm. So this in cytoplasm, what happens? So we have this kind of this enzyme. So in presence of this enzyme, so that enzyme gets oxidized, and this metal. So you have this metal. So that gets reduced to the M0. So this kind of the process happens in the cytoplasm. And this is called the intracellular synthesis of nanoparticle. Similar things happens here. If that enzyme is present there on the surface, so that metal ions get reduced into the metal nanoparticle on the surface. So here, that extracellular enzymes may be kind of the enzyme, maybe the protein, organic molecule solutions, the synthesis of particles. So these are the things may be present there. And we have the kind of things as fungus various types of the proteins, enzymes, nitrate reductase. So this can reduces metal and into the meat, and this for that leads to the formation of nanomaterial. And if you want to further understand the mechanism here, so in extracellular kind of the mechanism here, so what happens? We have the kind of the oxidoreductase kind of the enzyme. So in this presence of enzyme, so that metal and get reduced to the M0, and that gets oxidized into the this one. So if that oxidized to this one, so that get reduced, and for the this kind of the process happens by this cellular mechanism metabolism process. And if you want to understand the intracellular kind of the process happens, so here the metal ion that enters within the cell using that kind of the transportation mechanism. And with that metal ion gets reduced to this one in the presence of kind of this metabolic activity and the kind of the active detox detox species present in cytoplasm. Maybe this process happens. Maybe there is the kind of the enzyme process. Enzyme. So maybe the kind of the detox species, maybe in the presence of the enzyme. So these are the things happens within other cytoplasm, and finally we'll get this metal ions get converted into metal, and there is formation of the nanomaterial, and that is kind of the bioaccumulation material in the cytoplasm. So these are the differences in the intracellular and extracellular mechanism. And if you want to take the example, so for example here, that kind of the fungus is the Fusarium oxysporum. And so if we can synthesize go or silver nanoparticle using this Fusarium oxysporum, so this metal ion plus this one to gets reduced to the silver nanoparticle, and here on this Fusarium oxysporum, this kind of the enzyme is present. So that enzyme gets oxidized into the NAD plus. So we are again and again we have encountered with such kind of the enzyme, and that popular enzyme is called the NADH. So we need to just learn NADH is very important enzyme, and that is oxidized into the NAD plus, and that reduces Mn to the M0, and that leads to the formation of nanoparticle. So this kind of the mechanism we can use maybe the intercellular, maybe the kind of the extracellular kind of the mechanism. Now, we can also opt for the different kind of approach. And that kind of approach is that, suppose we have fungus here. So we can extract the meaningful and important chemical from that kind of the fungus. For example, so we can extract these kind of chemicals. Maybe we can extract the polysaccharides, enzymes, proteins, flavonoids, terpenoids, phenolic organic acids, or maybe alkaloids. So if we just extract these kind of the important chemicals, so these are called natural reducing agent. So in presence of that extract, if we add here that metal and metal ion solution in that extract, what happens? So since this is the strong reducing natural reducing agent, and the same time it is the stabilizing agent or maybe the capping agent. So in presence of this one, what happens? So this M plus that gets converted into the M0, and that gets the kind of this metal nanoparticle. And here that similar kind of that polysaccharides with enzymes also stabilizes that nanoparticles, and we have that stabilized nanomaterial. So we had under discussion about the use of sodium citrate. So we use citrate for synthesis of nanoparticles. Maybe we have used silver nanoparticles, gold nanoparticles, and we understand the citrate plays a role by so this is kind of the reducing agent. At the same time, it is kind of the capping agent, or maybe a stabilizing capping agent. Similarly, here this kind of the material this behaves as kind of the citrate where that we have such a reducing agent and a capping agent, and that stabilizes nanomaterial synthesis. Okay, so now let's understand the difference between the synthesis of nanomaterial by fungi and this bacteria. So here in fungi, so here if we try to develop the gold nanoparticle using fungus and bacteria. So whatever, there is metal toxicity resistant. So the fungus has the very high metal toxicity resistance. So what happens if that has a very high resistance? That means that converts more nanoparticles as compared to this bacterium. Second is kind of the location of that material. So in fungus, the most commonly that is the extracellular kinds of the location nanomaterials. However, in bacteria, maybe we have the intracellular, maybe the extracellular kind of the nanoparticle location. And the third important point is that in the biosynthesis rate. So in the fungus, we have the very fast synthesis of the nanomaterial. However, in this bacteria, we have the very slow process, and that may take more than 24 hours as compared to this fungus that maybe take less than 1 hour. So that's the important point here. So we can explain based on the kind of the synthesis rate. And here, and next point is the nanoparticles shape and size. So here shape and size depends on the biomass and the goal ratio as I explained. And in the case of the bacteria, we have the spherical kind of this nanomaterial synthesis. And here if you talk about the reducing agent, so we have the NADS kind of the reductase kind of the enzyme, or maybe kind of this phytogelatin or maybe melanin. However, in the bacteria, maybe we have this produce redox mediators, maybe the membrane protein or cytochrome, inorganic redox compounds. So these are those active bio-reducing agent in bacteria. And I think about the bio-capping agent for the stabilizing of nanomaterial. So here in this, there is kind of the identification of that surface. And so in this bacteria, still we don't get kind of the specific agent, but it is reported in some literature that cysteine, tryptophan, and tyrosine, so these can be used as the kind of the capping agent in the gold nanoparticle synthesis. And if you think about the use of this kind of biological sample at the industrial scale, So we can use the fungi kind of the system for the nanomaterial synthesis more likely as compared to the bacteria because the rate of the nanomaterial synthesis. Okay, so today we learn mechanism of the nanomaterial synthesis using bacteria and the fungi. And tomorrow we will learn nanomaterial synthesis using protein and the kind of the biotemperature nanomaterial synthesis. So see you in the next class. Thank you.